Welcome to the Passionate Minds course content review. Today we'll be covering proteins and nucleic acids, and you'll learn about their properties and roles within biological systems. So nucleic acids are biopolymers that are essential to life and contain hereditary information. They're macromolecules that are made out of units that are called nucleotides. And they come in two naturally occurring varieties. So you have DNA, which is deoxyribonucleic acid, and you have RNA, which is ribonucleic acid. So DNA is the genetic material that is found in living organisms, and that goes all the way from a single-celled bacteria to a multicellular mammal. So the structure of a nucleotide, which is the monomer in this case, it's made up of three different parts. So you have a nitrogen-containing ring structure called the nitrogenous base. Then you have the 5-carbon ring, and then you have the last part, which is a phosphate group. So the sugar molecule has a central position in the nucleotide, and then you have the base that is attached to one of its carbons, and the phosphate group is attached to another carbon. So nitrogenous bases of nucleotides are organic, so these are carbon-based molecules that are made up of nitrogen-containing ring structures. So why do we call them nitrogenous bases? It's because some of the nitrogens in a base could be protonated, so they could pick up an H+. So what will happen is this will then decrease, it will lower the hydrogen ion concentration, which means that it is a base in the acid-base sense. Okay, so each nucleotide in DNA contains one of 4 possible nitrogenous bases. So you have adenine, you have guanine, you have cytosine, and you have thymine. So these are represented by letters A, G, C, and T. Adenine and guanine are purines, which means that they are double ring structured. So they have 2 fused carbon-nitrogen rings. Cytosine and thymine are pyrimidines and have a single carbon-nitrogen ring. So the way I remember this is that for purines, marriage is pure, so you have 2 rings. You have a marriage amongst 2 people, so you have 2 rings, and usually rings are composed of silver, so Ag. So that's the chemical formula for silver. So you have 2 silver rings in a marriage that is pure. So that's how to remember purines. For pyrimidines, I just remember cut, and I automatically think of pyramids for some reason. That's how I personally do this. But I also have to consider one more thing too. RNA nucleotides, they bear adenine, guanine, and cytosine bases, but instead of thymine, they have another pyrimidine base that is called uracil. So now we see uracil over here. RNA has uracil, but it doesn't have thymine, and DNA has thymine, but it doesn't have uracil. Also, another thing to consider here too is that G's always go with C. So cat goes in the garage, and your A's always pair with the T. And in terms of this over here, we have to consider the numbers of hydrogen bonds that form between each of the pairs. So when you have guanine and you have cytosine on the other side, you form 3 hydrogen bonds. When you have adenine and you have thymine, you form 2 double bonds. This occurs when you have a double-stranded DNA helix. Now let's talk about the differences between DNA and RNA. I sort of mentioned a little bit in the previous slides, but I'll be more evident in these ones. So the function of DNA is it's a storage of genetic information, Whereas in RNA, RNA is involved in protein synthesis and gene regulation, and it is a carrier of genetic information in some viruses. In terms of the sugar for the 5-carbon sugar for DNA is called deoxyribose, whereas in RNA it is ribose. So the difference between deoxyribose and ribose is the presence of oxygen in carbon 2. So we see over here you have a hydroxyl, but over here you just have a hydrogen. So in deoxyribose, without oxygen, you just have the hydrogen, and in ribose, you have the hydroxyl here on carbon 2. The structure of DNA is in the form of a double helix, whereas in RNA, it is single-stranded. Single-stranded. So DNA is double helix, and RNA is single-stranded. The bases involved in DNA are cytosine, thymine, adenine, and guanine, whereas in RNA it is cytosine, uracil instead of thymine, adenine, and guanine. So RNA is made in the nucleus of the cell, and when it is transported into the cytoplasm, it is essential in the process of transcription, which we'll talk about more when we get to molecular genetics. So a nucleotide strand is formed when a series of nucleotide monomers bind through the formation of phosphodiester bonds. So we see an example of a phosphodiester bond over here, where phosphate is linking these two nucleotides. So we see that nucleotides may have a single phosphate group or a chain of up to 3 phosphate groups that are attached to the 5' of the sugar over here, the 5' carbon of the sugar. But in a cell, when a nucleotide is about to be added to the end of a polynucleotide chain, it will bear a series of 3 phosphate groups. But when a nucleotide joins the growing DNA or RNA chain, it will lose those 2 phosphate groups as a result of a condensation reaction, and thus result in the formation of a phosphodiester bond. In a chain of DNA or RNA, each nucleotide will just have a single phosphate group, as shown over here. So you just see one phosphate group in each nucleotide. Another important thing to consider is that both DNA strands are anti-parallel to one another. So one strand will begin with a phosphate group from the 5' carbon, whereas another on the opposite end will begin with a sugar on the 3' carbon. So this is an example of a double-stranded DNA here. And like I mentioned before with the hydrogen bonds, remember that A and T have 2 hydrogen bonds and G and C have 3 hydrogen bonds between them. So this is just a double-stranded DNA. So proteins are one of the most abundant organic molecules that are in a living system and are very, very, very diverse in structures and functions compared to any other macromolecule that we'll talk about, that we've already talked about. A single cell can contain thousands of proteins with various functions, and although their structures, like their functions, could vary greatly, all proteins are made up of one or more chains of amino acids. So they have many roles. They could either catalyze chemical reactions, they provide structural support, they can transport substances, they enable organisms to move, and they regulate various cellular processes. Another thing too to mention is that they can also defend against diseases. So in this diagram that I've pasted over here, it shows that the structure differs between every protein, therefore it can undergo many different functions within the cell. So amino acids are the monomers that make up proteins. Specifically though, a protein is made up of one or more linear chains of amino acids. So this one or more linear chain of amino acids, it is denoted as a polypeptide. And so there are 20 types of amino acids that are commonly found in proteins. So amino acids, they share a basic structure in this diagram here that I've noted. So you have a central carbon atom that is denoted as the alpha carbon, and then you have it bonded to an amino group like we looked at with the functional groups. You have a carboxylic acid group on the other side, and then you have a hydrogen atom that is linked to the alpha carbon. This R group is a side chain, which I'll talk about in a bit, but let's talk about the structure a bit more. So we have a polypeptide chain over here, and so you have an N terminus and you have a C terminus. So the N terminus refers to the side with the amino group, and the C terminus refers to the side with the carboxylic acid group. Every amino acid also has another atom or group of atoms bonded to the central atom, and that is known as the R group. So the R group over here. So the R group is what determines the identity of the amino acid. It'll determine its properties and characteristics. So if the R group is a hydrogen atom, that means that the amino acid is called glycine. If it's a methyl group, that means it's called alanine. So there's 20 common amino acids, and I'll show you them in the following slides. I'll talk about whether they're electrically charged, they're nonpolar, or they're polar. And they're highlighted more clearly. I'll go through them. So nonpolar amino acids have little or no polarity in their side chains. Like we see over here, there's a minimal electronegativity difference, and there's no dipoles. Even if there is a dipole, like we see with the nitrogen and tryptophan, they're there, it's offset by the large R side chain. And so this lack of polarity overall means that there's no way to interact with the highly polar water molecules, and therefore they're hydrophobic. Now, a way to memorize these is to use the mnemonic or acronym GLAMP TVIP. So GLAMP TVIP, nonpolar hydrophobic, so you have glycine, leucine, alanine, methionine, phenylalanine, tryptophan, valine, isoleucine, and proline. So polar amino acids are hydrophilic, which means that they have polarity in their side chains. So this polarity means that they have a way to interact with highly polar water molecules, and thus they're hydrophilic. So this is because of either the short R chain or the dipoles that are present, with the high electronegativity difference, and thus it makes it polar. A way to memorize this is to use the acronym, or the acronym GAS TTC, where you have glutamine, asparagine, tyrosine, threonine, cysteine. Sorry, I forgot to mention serine over here too. In terms of the electrically charged amino acids, we have acidic, which are negatively charged. The reason why they're negatively charged is because they donated a proton. And for the other three, these are basic because they're positively charged, which meant that they accepted a proton. So the way to remember the three basic amino acids is to use hallelujah. So the first two letters of hallelujah are HAL, so that represents histidine, arginine, and lysine. For acidic, these two, there's just two to remember, so it's not that difficult. So glutamate and aspartate, or aspartic acid, or glutamic acid. So now let's talk about the protein structure. So there's 4 levels of protein structures. There's primary, secondary, tertiary, and quaternary. So in a primary protein structure, it is the sequence of amino acids. So it's like the order of letters in a long word. The amino acid sequence determines the overall function of the protein. It influences the final conformation of the protein, so how it behaves, its characteristics, and its functions. And it is determined by the inherited genetic information. So this we'll talk about more in the molecular genetics unit. And also another thing to mention is that there's 20 to the n possibilities in a protein. So n is the number of amino acids in this case. Now let's look at this diagram over here. Uh, this is a polypeptide. So you have a polypeptide formation that's occurring over here. So you have between each individual amino acid, you have a peptide bond that is formed. And this is formed through a condensation reaction. So we see that water is being released over here. And eventually these guys will become linked. So the secondary structure is a result of hydrogen bonds that form between the peptide backbones. So this does not include the R groups. So what happens is as the chain grows, it coils and folds at different locations along the length of the peptide backbone. So hydrogen bonds are therefore formed between the amino acids at different locations in the polypeptide chain, and that will help to maintain the structure. So you have two secondary structures. So you have an alpha helix and you have a beta pleated sheet. So this is an alpha helix and this is a beta pleated sheet. So let's talk about an alpha helix more in depth. So in an alpha helix, what happens is you have the carbonyl of one amino acid interacting with the amino portion of another amino acid. And you have a hydrogen bond that is formed between them. So this hydrogen bond will pull the polypeptide together into a helical structure that resembles a curled ribbon, like you see here. It's like a curled ribbon. And what will happen now is that each the R groups of the amino acids will then stick outwards from the alpha helix, where they're free to interact with each other or with other molecules. In a beta pleated sheet, however, you have two or more segments of a polypeptide chain which line up next to each other, and that will form a sheet-like structure that is held together by hydrogen bonds. So the hydrogen bonds form between carbonyl and amino groups of the backbone. So we see over here there's an amino group here and there's a carbonyl group here. So they form the hydrogen bonds in that direction, while the R groups extend either above or below, below the plane of the sheet. Certain amino acids are more or less likely to be found in alpha helices or beta pleated sheets. For instance, we see the amino acid proline. Proline is sometimes called a helix breaker because of its unusual R group. So in this case, it'll bond to the amino group to form a ring and create a bend in the chain, which is not compatible with the helix formation. So proline is typically found in bends and unstructured regions between secondary structures. Amino acids like tryptophan, tyrosine, and phenylalanine have large ring structures in their R groups and are therefore found in the beta pleated sheets because the beta pleated sheet structure provides plenty of space for the side chains. So the overall three-dimensional structure of a polypeptide is called its tertiary structure. This tertiary structure is primarily due to interactions between the R groups of the amino acids that makes up the protein. So you have R groups that are mainly present here that contribute to the tertiary structures. So these interactions can include hydrogen bonds, ionic bonds. You have the van der Waals forces, which includes the dipole-dipole, London dispersion forces, and the hydrogen bonds. So let's look at this diagram over here. So R groups with like charges repel one another, whereas those with opposite charges can form an ionic bond. Similarly, polar R groups form hydrogen bonds in other dipole-dipole interactions. But it's also important to note that the tertiary structure includes hydrophobic interactions too, in which amino acids with nonpolar hydrophobic R groups will cluster together. They'll interact with each other and they'll be kept in the internal portion of the structure. Whereas hydrophilic polar amino acids are on the outside and they want to interact with the surrounding water molecules. Finally, there's one type of covalent bond that can contribute to the tertiary structure, which is special, and it's called a disulfide bond. So disulfide bonds are covalent linkages between the sulfur-containing side chains of cysteines, so the cysteine amino acids, and they're much stronger than other types of bonds that contribute to tertiary structure. So they act like safety pins, which keeps the polypeptide firmly attached to one another. A quaternary protein structure refers to two or more polypeptide chains that come together to form one large macromolecule. So this over here is hemoglobin, this molecule right here. This is hemoglobin, and it consists of 4 polypeptides, so 4 subunits. So you have 2 alpha subunits, and you have 2 beta subunits here. So many proteins are made up of a single polypeptide chain, and they'll only have 3 levels of structure. So the ones we've already discussed, primary, secondary, tertiary. But some proteins like hemoglobin have multiple polypeptide chains, and these multiple polypeptide chains are known as subunits. So when these subunits come together, then they'll give the protein a quaternary structure. Hemoglobin, its role is to carry oxygen in the blood, and it is made up of 4 subunits. So you have 2 alpha subunits and you have 2 beta subunits. But in general, what happens is the same types of interactions that contribute to tertiary structures. So these are like weak interactions such as hydrogen bonding and London dispersion forces. These will hold the subunits together to give the quaternary structure. So London dispersion forces and hydrogen bonding helps to give the quaternary structure. So in addition to primary structure, physical and chemical conditions can also affect protein structures. So for example, alterations in the pH concentrations, you have salt concentrations, temperature, or other environmental factors that can cause a protein to denature or unravel. So denatured proteins are biologically inactive, and improperly folded proteins are implicated in many diseases. So I didn't put the example here, but Alzheimer's and mad cow disease, they occur because of improperly folded proteins. Here in this example, we see that heating an egg to extreme temperatures can lead to irreversible denaturation of the egg protein. So the egg protein here is called albumin, and that's what makes the egg go from liquid to solid because of irreversible denaturation. In this diagram over here, what we see is that a change in the primary structure can affect a protein structure and function. So sickle cell disease is what results from a single amino acid substitution in hemoglobin, which will result in the structure of the protein changing and the cell as a whole. So the cell will become very non-functional. So that is what you call sickle cell disease. In terms of this, I wanted to highlight this point again here. Protein folding relates to where we can or will find a protein within the cell. So like I said before, polar side chains on the outside of the molecule so that they can form hydrogen bonds to water. But in the interior component, you have a hydrophobic core, which contains the nonpolar side chains. So enzymes catalyze chemical reactions. So there are proteins that can speed up or catalyze chemical reactions, and they function at an optimal pH or temperature. This means that they could be denatured if they're not in an ideal environment. So these are catalysts for biochemical reactions that happen in living organisms, but sometimes they could be RNA. They could be ribonucleic acid molecules that also can be behaving as enzymes. So enzymes, they perform the task of lowering a reaction's activation energy. So the activation energy is the amount of energy that must be put in for a reaction to begin. So enzymes will work by binding to the reactant molecules, and then it can hold them in such a way that the chemical bond breaking and bond forming processes take place more rapidly. So this is the transition state, an unstable state where products must pass through in order to become reactants. So to catalyze a reaction, what will happen is an enzyme will grab on to one or more reactant molecules, and these molecules are known as the enzyme substrates. So one substrate can be broken down into multiple products, but in another enzyme catalyzed reactions, two substrates can come together to form one larger molecule, or to swap places. So whatever biological reaction you could think of, there is probably an enzyme that can speed it up. So the part of the enzyme where the substrate binds to is called the active site. So that is where the catalytic action occurs. Instead, an enzyme will change shape slightly when it binds to its substrate over here, resulting in an even better fit, induced fit. So this adjustment of the enzyme is necessary, and that is what you call the induced fit. So let's go through each of these steps here. So this is when the substrate enters the active site of the enzyme. And then what happens is you'll have the enzyme substrate complex in step number 2, where the enzyme will change shape slightly as the substrate binds. In the third step, the enzyme this is called the enzyme products complex. And then in the fourth step, the shape of the enzyme reverts back and the products leave the active site of the enzymes. So what does the enzyme do to lower the activation energy? So it depends on the enzymes. They could either speed up chemical reactions by bringing two substrates together in the right orientation, or they can create an environment inside the active site that is favorable for the reaction. So one that could be slightly acidic or nonpolar. As we saw with the R groups, there could be acidic or nonpolar. The enzyme substrate complex can also lower the activation energy by bending the substrate molecules in a way that will facilitate bond breaking, and this will help reach the transition state. Or enzymes could also lower the activation energies by taking part in a chemical reaction themselves. So the active site residue can form temporary covalent bonds with substrate molecules as part of the reaction progress. So this is temporary. An enzyme will always return to its original state at the end of the reaction.